Okay, hi guys. So yesterday we did transcription, and today we're going to do translation. Remember, these are the two parts of protein synthesis. Transcription happened in the nucleus when you're making the mRNA copy, and then that mRNA, once it's processed and has the 5' GCAP and the poly A tail, can go out into the cytoplasm. Its ends will be protected, and then meet up with the ribosomes and do translation. So here's what we're going to talk about. Just to review, so the rounded ones in your kit, that's DNA. The square ones in your kit are RNA. So here's the 5' end with the indentation of the arrow. Here's the arrow sticking out, and that is the 3' end of those sugars. This is the sequence of the DNA. You can automatically recognize it's DNA, even though it is single-stranded DNA, ssDNA, because there's the presence of the T. So the corresponding complementary strands, because remember your DNA is complementary, so A pairs with T, C pairs with G, and they're going to form hydrogen bonds with each other. Just kind of a quick review when you're putting together your DNA replication videos. Also, what you're going to be doing is creating a transcription translation video. So once you finish viewing this, you'll have all the information you need to start putting that together. And that's not due till later in the month. Transcription of DNA into RNA. So this is happening inside the nucleus, because remember, your DNA stays nice and protected inside the nucleus. And this big red bubble right here is representing the DNA polymerase that's going to break apart, pry apart the DNA, those hydrogen bonds right there, and split it. And it's only going to need to read one copy of the DNA because that mRNA is single-stranded. So one of these is going to be the template strand, the sense strand, the non-coding strand, whatever you want to call it, the parent strand. That's what it's going to be. There's several names for it depending on what video you watch or what book you read. All right, RNA polymerase is going to build that mRNA sequence one nucleotide at a time. Remember, in mRNA, they don't have thymine, they have uracil instead, so that A codes for U. In the mRNA. And as that mRNA builds in the active site, as the RNA polymerase builds those complementary RNA nucleotides, it's going to actually branch off and kind of hang out of the DNA. And what that does is that allows the DNA to recoil. So that allows those hydrogen bonds to reform once the RNA passes by. All right, so here we go. mRNA codes for proteins in triplets. If you remember, we already talked about that. So in DNA, those might be referred to as triplets. In RNA, they're going to be referred to as codons. And so we usually have a reading frame where we break it apart into 3 nucleotide sequences. And so this then becomes our reading frame for the RNA polymerase. And then once the RNA polymerase makes the mRNA, each one of these codons is going to be read by the ribosome and is going to equate to one amino acid or a stop codon, which is going to initiate that ribosome to break apart because you've completed your polypeptide sequence. Okay, so each one of these codons, these 3-letter sequences, 3 nucleotide sequences, are going to code for an amino acid. And then those amino acids are going to be bonded together in polypeptides and form a polypeptide chain. Okay, so we are cooking. Here we go. Remember, we already talked about in class, the triplet code of the DNA will determine the codon of the mRNA. Here's an example of a codon wheel. So this is how you know which amino acid, which nucleotide sequence, which codon sequence codes for which amino acid. This will always be given to you on the AP exam. You will either be given the codon wheel or you will be given a codon chart. Okay, either one, and we'll practice kind of reading these in class. And you should also have these. This should have come in your kit. So, and I'm going to post a photocopy of them to the topics page, so you'll have that as well, just in case it didn't come with it. So the triplet code in the DNA is going to equate to a codon in the mRNA. That's the 3 consecutive nucleotide bases that specify one of the amino acids. We did talk about how you have the 4 possible nucleotides, A, C, U, G. And in a 3-letter combination, that is 64 possible code words or codons that can code for the amino acids. But there are only 20 amino acids, so this allows for redundancy. One code for each 20 amino acids, but you have repeats. So if you look right here for the leucine, you start in the center and you go CUU, codes for leucine. CUC also codes for leucine. CUA codes for leucine, and CUG codes for leucine. So you can see how this would be advantageous, because if you have a mutation or a base point mutation, so 1-letter change, in that 3rd spot in the codon, it's going to code for the same amino acid in the protein. So a mutation in your mRNA or mutation in your DNA that causes a different nucleotide to be placed in that 3rd-letter spot in your mRNA may not actually negatively impact the shape or structure function of the protein. So 61 of the 64 triplets code for actual amino acids. You do have stop codons, so UAA, UAG, and then UGA are your stop codons. And that's going to trigger the ribosome to stop reading the mRNA, dissociate, and your polypeptide is done being built. The 3 codons signal for the termination of the translation. Here they are again: UAA, UAG, and UGA. So the codon AUG, here we are, AUG, is going to code only for methionine. That's the only one that codes for methionine. All of your proteins start with methionine. All of your polypeptides start with methionine because this AUG is the start codon. That's what initiates the small subunit of the ribosome to bind to that mRNA at that point. And then the large subunit will attach, and then you will initiate the beginning of protein synthesis or building that polypeptide chain. All right, so we just talked about if you have a point mutation or that change of that 1 letter in that 3rd spot, we call that a wobble. So the 3rd position in the codon is a wobble. So there are 45 different tRNAs, those transfer RNAs that can come pick up those amino acids and bring them to the mRNA at the ribosome. That 3rd nucleotide pairing is less specific than the first 2, because you can kind of see how phenylalanine, leucine has 6 different codons that code for it. Isoleucine, you can have a U, you can have a C, you can have an A, and it doesn't really matter in that 3rd spot. It'll code for the same amino acid, and we call that a wobble. So that's that redundancy. Also, having the 45 different tRNAs, because of that wobble, that allows some tRNAs to be reused, and they can go back and pick up with that anticodon. They can just go back and pick up, like if you have a UU or a CU, they can pick up a leucine. It doesn't matter what that 3rd letter is. So it can pick up that specific amino acid. So the first 2 nucleotides in glycine's codon. So if you were to look for the GLY in glycine over here, it's GG. And it doesn't matter whether it is a U, a C, an A, or a G. In this genetic code chart, you're really going to look for the first letter is in like this right here. So these are the first letters, these are the second letters, these are the 3rd letters. So first letter is a U, second letter is a U, and then each one of these boxes. So that's how you read this square chart. On the AP, I do believe it even tells you first letter, second letter, 3rd letter. All right, so splice and process mRNA, because remember that mRNA, when you make a copy of it from the DNA, is going to have introns and exons. So if you remember, the way to kind of remember it is that the introns, the non-coding regions, are going to stay in the nucleus. They're going to be spliced out by the spliceosomes, the SNRPs, and stay inside the nucleus. The actual coding region of those genes are the exons, and they are going to exit the nucleus. They're those pieces that the spliceosomes splice together, and they make the mRNA, the message. So the spliced and processed mRNAs then transfer to the cytoplasm, to the ribosome, and the next part is called translation, because remember, we're going from the language of nucleic acid, that mRNA, to the language of amino acids, because we're going to be building a polypeptide chain that eventually will make proteins. So the corresponding polypeptide for this mRNA sequence, assuming no poly A tail or 5' GCAP, is we're going to look at this. So here we have AUG, which on your chart Okay, so looking at your chart, we have A, U, G is going to code for methionine. So we know that that's our first one. And remember, you're going to break these up into a reading frame of 3 nucleotides at a time. Okay, so we should have 1, 2, 3, 4, 5, 6. If these are all, because these are processed, so these are all exons, they've been spliced together. So we should have 6 amino acids in our chain. Okay, so we have methionine, proline, tryptophan, isoleucine, and glutamate. All right, so it's like 1, 2, 3, 4, 5. And it's like, all right, why do we only have 5? Well, if you look, UAG is actually what codes for a stop codon. So here's our start codon, AUG, and here's our stop codon. Stop codons do not code for a amino acid. They actually will code for a modified tRNA that does not attach to an amino acid to come, and it lands in the A site of the ribosome and signals the ribosome to dissociate the large subunit and the small subunit. So they'll break apart, and that will terminate translation. And that's the whole point of a stop codon. All right, so that tRNA, kind of the little modified stop codon, we also have these. So if you want to use these for your video, I'm going to upload a photocopy of what these look like, or you can hand draw them, use little sticky notes. You know, they kind of sort of look like a T. But basically what happens is the amino acid is going to attach on the 3' end or up here. And each one is going to specifically pick up an amino acid that corresponds to its anticodon. So remember, we did talk about this. The anticodon is going to be complementary to the codon on the mRNA. So that's right here. And this is how that tRNA knows where to attach. So if its anticodon does not line up with, if it is not complementary to that mRNA, it is not going to release its amino acid into the polypeptide chain. It's going to leave the A site of the ribosome and another tRNA will come in. So the tRNA with the appropriately bonded amino acid at the top will land in the ribosome. It will line up its anticodon, its complementary anticodon, with the mRNA codon down below. And remember, these are both mRNAs. Scratch that. These are both RNAs. So C codes for G, G codes for C, A codes for U, and U codes for A. I don't have any T's in there. All right, there is an enzyme that you have in your cytoplasm, the aminoacetyl tRNA synthetase, that is going to actually attach it's responsible for attaching the appropriate amino acid to the correct tRNA group on the 3' end of the tRNA. This enzyme does use energy. It uses 2 phosphate bonds from ATP, so it turns adenosine triphosphate into adenosine monophosphate. So it does require a lot of energy. However, the bond between that amino group right here, that amino acid, is unstable. And that's actually really helpful because you want it to be bonded to the tRNA temporarily, because really this is just to transfer, hence the tRNA name. It is to transfer the amino acid from the cytoplasm specifically to the ribosome so that it can then line up in the correct order and form the polypeptide chain. So you want it to be able to readily release that amino acid when it gets to the ribosome to form that peptide bond. All right, so you have 3 spots that are in the ribosome. So you have the aminoacetyl tRNA site, so that's the A site. You have the peptidyl tRNA site, that's the P site. You can think of also the P site as the polypeptide chain site. So this is going to be the site where the actual polypeptide chain is formed. The tRNA that's going to carry that growing polypeptide is going to be in the P site. And the E site is the exit site. That's where a tRNA that doesn't have an amino acid is going to move over to the E site and then exit the mRNA. Oh my goodness, it's going to exit the rRNA, the ribosome. Whew. That's a rough one. So what happens is you have a small subunit down here. You have the large subunit up here. Here is your mRNA strand, okay? This is the message being read. So if this were the first message that's coming in, then this would be your AUG, your start codon right here, your anticodon to AUG. Remember, A codes for U codes for A, and then G codes for C. So your anticodon would be UAC, and then it would carry methionine. So the amino acid methionine, which is abbreviated MET. Okay, so that would be like this would be your MET. Your anticodon right here would be your UAC. Kind of space them out so they fit right there. Because your start codon on your mRNA would be AUG. Okay, it's kind of hard to see in the blue, sorry. And so your first tRNA is going to land in the P site. Your second tRNA is going to come in and land in the A site. So the ribosome assembles and initiates at the P site. So it assembles around the start codon at the P site. The first amino acid comes in. The second tRNA with the amino acid comes in. As the ribosome shifts down, this tRNA will donate, release, because this is an unstable bond right here, its amino acid to the tRNA from the A site, because the tRNA that's in the A site will shift over to the P site. And then the one in the P site, which now has given up its amino acid and formed a peptide bond with the one in the A site as it shifts down, will then move to the exit site and leave. A new one will come into the now vacant A site, and the tRNA that's in the P site should have two amino acids on it, the one from methionine and the one it was also carrying. So that's how the polypeptide chain builds in the P site. It's as the ribosome shifts down reading frames. So as it shifts down codons, a new tRNA is going to come into the A site, and that tRNA that's vacant is going to leave the E site, the exit site. All right. So there are three phases, and you do need to be familiar with these terms for the test, for your homework, and also for your videos, and also for the AP. And we do break up transcription and translation into three phases. We call them initiation, elongation, and termination. So initiation is going to basically bring the mRNA, the ribosomal subunit, that small subunit, and that large subunit together with the initiator tRNA. So the initiator tRNA is the one that is carrying the methionine, that start codon. Elongation is basically as you add those amino acid bases to the codon sequence. So as the new ones come into the A site, it shifts down, and you build that polypeptide chain by adding more peptide bonds between the amino acids. And then you're going to reach termination once the stop codon is reached, because remember at the stop codon, you have a release factor. So it's a modified tRNA. It doesn't bring an amino acid. It just signals that it's time for the large and the small subunit to dissociate and break apart. And then once that happens, this tRNA is going to release the amino acids as it shifts to the E site. It will go off into the cytoplasm, go pick up another amino acid, because some of the ribosome is also making another polypeptide chain. Excuse me. And then your polypeptide is then released. And so it can fold, or it can meet up with other polypeptides and be modified into a protein. So this is one thing that I do want to talk about, and I kind of want you guys to see. So translation continues codon by codon, and this is really the progression as it goes. And yes, it does require energy in the form of GTP. So we did talk briefly when we talked about DNA replication, that all of those nucleotides, that adenine, the adenine nitrogen base bonded to that 5-carbon sugar, that pentose sugar, makes up adenosine, and then bonded with one phosphate group, that's adenosine monophosphate. And so we talked about how all of your nucleotides actually arrive as ATP, GTP, CTP, and TTP. And they bring with them in those two extra phosphate bonds the energy to create those phosphodiester covalent bonds that form the backbone of your DNA. Well, the same thing happens with the nucleotides that come in for making that new mRNA sequence. So only they're going to be UTP instead of TTP. So there's a lot of energy used. You also use GTP and convert it into GDP. So you're using one of those phosphate bonds, you're breaking one of those phosphate bonds, but you're using two of them at a time. And that's utilized by the ribosome to make that peptide bond between one of the amino acids and the growing polypeptide chain. So to add those two together, and then also to shift the reading frame down. So again, adding more on, shifting it down, it takes energy, right? So this whole process utilizes GTP energy. So remember, we talked about how you have ATP, CTP, TTP, GTP, UTP. Those are all forms of energy that your body can utilize. We just mainly talk about ATP formation with the mitochondria. All right, so codon by codon, it's going to add down and you're going to create that chain. And it talks about how here's the recognition of that first codon. And then another tRNA is going to come in. It's going to bring its amino acid that's not very stably bonded to the tRNA. It's going to utilize energy to form that new peptide bond. It's going to increase the size of that polypeptide. That amino acid, that tRNA, the amino acid chain is going to temporarily be attached to the one that's in the A site. But remember, the ribosome is just moving down the reading frame, so it's shifting. And so this polypeptide chain right here attached to this transfer RNA is going to move to the P site. The tRNA that doesn't have an amino acid attached to it is going to move to the exit site, and then eventually it's going to leave. The A site's now vacant, and then that's where the new tRNA, the new transfer RNA with the amino acid attached, is going to land. And so this is basically the phase. It just does. Now, I do want you to see that you can actually form something that we call polyribosome chain. And this is actually how a lot of your proteins are made. It doesn't really do you any good to make one tiny polypeptide strand, especially if your body is attempting to make proteins for export or make proteins inside the cell and you need a large reaction. So remember when we talked about cell signaling and ligands, and proteins can be parts of those. And you can have autocrine signaling and paracrine signaling. So remember those. And proteins can be those signals. And so you need lots of those proteins, higher concentrations of those proteins. And so what you really need is one mRNA sequence, and it's read by several ribosomes at once in series. So as one ribosome moves down, it makes room for another ribosome to then land at that initiation complex, at that start codon, and then to start reading down. And so you do have this continuous stream, and we call this the polyribosome. And here's this growing polypeptide that you can see right here, and eventually it'll be released. Let me clear some of those drawings off there. My apologies. So this actually is electron microscope of a prokaryote. This is one in a prokaryote, but it's very similar in eukaryotes. We do this as well. So each one of these spots right here, kind of these circles right here, are ribosomes, and they're reading one mRNA sequence. So I want you guys to understand that it's not that you make one polypeptide like one and done. You are making lots of polypeptides, pretty much all sequentially, because your body went through the energy, it used the resources to make that mRNA. And so it wants to maximize its output from that input, basically. It wants to get as much out of that code for those genes as it possibly can. And eventually, if it doesn't need it, it'll turn it off. All right. When a stop codon is reached, the polypeptide is then freed. It's let go. So here's that releasing factor that's going to bind into the A site. And then that tRNA is going to release the polypeptide chain. It's going to cleave it, and then it's going to dissociate. It's going to leave. It no longer has an amino acid attached to it or polypeptide chain attached to it. And then the large subunit and the small subunit of the ribosome are going to release. The polypeptide may undergo folding, cleaving. It may be added to another group. It may have a glyco, so sugar, or a lipo, fat group, lipo group added onto it, depending on whatever function it has. So it's going to go on to be modified, but it's not quite a protein yet. If you remember when we covered macromolecules way back when. With proteins, you have primary proteins, which is that polypeptide chain, that amino acid sequence. That's a primary protein. It's not a functional protein yet. Collagen can be a functional secondary protein if you link several of those secondary chains together. So if you remember that, the alpha helices and the beta pleated sheets. Then in the tertiary structure is where those functional R groups and R group interactions come in handy to form more 3D shapes or folding in a protein. And then if you remember the quaternary structure, protein was the more globular protein, and you can attach several tertiary globular structures together. And that's how you form things like hemoglobin that actually has 4 subunits. All right. Now, your body can have two things because remember, you have ribosomes attached to the endoplasmic reticulum, that rough ER. Those are ribosomes. You also have free floating ribosomes. And we talked really briefly about the fact that you have, if you have free floating ribosomes and that's what reads the mRNA, that's going to be for internal use inside the cell. But if you are reading the mRNA in a ribosome that is going to be attached to the rough ER, that is going to be a protein for export. And so protein targeting, because remember, if you're going to export the proteins, you also need to tag the proteins. So you have these things, these signal recognition particles, SRPs, they can be another type of RNA. So this is in the cytosol, so the cytoplasm. Your ribosome, this is the mRNA. It's reading that code. And basically, when it attaches that SRP, that's kind of the address label, the shipping label. And the destination can be, it can be secreted. The nucleus can go to the mitochondria, chloroplast, cell membrane, cytoplasm, wherever it is, it's going to be tagged. And then the ribosome, as it reads it, will actually insert. So this SRP, this label actually is read by a recognition complex, a translocation process that opens up the ability for that ribosome to insert its growing polypeptide chain into the ER. And then once it has it, the protein can be formed, and that protein then can be mixed with other proteins. It can be packaged up in a vesicle to go to the Golgi body for export. It can be tagged for internal use. It can be modified again with adding a glyco group or a lipo group. So the endoplasmic reticulum can do that. All right, and then this is how your kit will look. So I'm going to add a photocopy of the ribosome that you can actually use for your video. You're going to make an mRNA sequence. So we're going to give you some amino acid, an amino acid sequence, and then you're going to have to then kind of go backwards, amino acid to mRNA codon. And this is how you would show the translation, not transcription, translation. So basically, here's your AUG. This actually should be on the P site. But it's going to initiate. This is the tRNA is going to land. You have the methionine. You have a new tRNA hanging out. Remember, we did talk about your 45 tRNAs. They're floating around in the cytosol and the cytoplasm outside the nucleus, just hanging out. You have amino acids that have been recycled, that you've gotten through digestion and production, and they're hanging out in the cytoplasm, the cytosol. And you have lots of ribosomes that are also coming in to do this. So here we go. So kind of more progression along, the ribosome is going to read from the 5' to the 3' end. It's going to flow along, and you're going to have the tRNA with, I wanted you to see the letters, by the way, for the different sites. And so here's one that doesn't have the amino acid attached to it. So it is going to exit. The growing polypeptide chain is going to be built on this P site tRNA. And then as it shifts down, remember, you're going to use GTP energy to attach this amino acid to this growing polypeptide chain. And it's going to come out of the ribosome. And this ribosome is going to shift down 3 codons at a time. That's the reading frame of the ribosome. And remember, the tRNA knows where to land in the new A site, this new incoming site, because its anticodon is complementary to the mRNA's codon. So U and A, C and G, those are complementary. Okay. Eventually, so UAG, you're going to reach a stop codon. That is where you're going to get the termination of the sequence. As it shifts down, the tRNA is going to release this amino acid polypeptide. polypeptide chain. And then the tRNAs are going to release and leave out the exit site, and the small subunit and the large subunit are going to dissociate. And that is pretty much it. What I would recommend is that you watch videos for this process. And I'm going to post that video that we did watch in class with the real-time DNA replication. And I definitely want you to also watch the real-time transcription translation, because it's very, very cool to see that animation. Let me actually see if I can find it. Let's go replication, real-time animation. See if it's on YouTube, and I can find the one I like. Oh, there it is.
Grammarly can help you write quickly and
everyone loves Grammarly. Okay, so this is the one that kind of has the weird sound. So it's like, here's the human cell, 10 times magnification. Here are your histones. Here's your supercoiled DNA as it wraps around the histones. Remember, real quickly, we talked about how that's you turn on and turn off certain chromosomes by tightly coiling. Here's gene transcription. So here's these ribosomes reading mRNA. And then we're going to go down, let's see, and talk about, I'm going to scroll past the supercoiling of the chromosomes. Here we go. So here's that DNA replication. Remember, we talked about in real time, here's that leading strand that's coming off. And here's that lagging strand that's going off to the side. So the old DNA right here is the parental strand, and then the new daughter strands are being added on so that you get two separate sequences. So here it's adding that primer at that yellow, and then the DNA polymerase is moving off to the side. And so you get those Okazaki fragments, because remember, the DNA polymerase only reads directionally. All right, so I'm going to skip past the DNA replication because we already did that. And then here is, okay. So what's happening right here is this is the promoter region. Vitamin D is going to be a cofactor that's going to help initiate this factor at the promoter region. Your RNA polymerase is going to bind with the help of these promoters and transcription factors. So these are, there it is. So here's your RNA polymerase that unwinds as it goes and is building the mRNA. So this is transcription happening in the nucleus. So this is real time, by the way, so that's how fast it happens. So here's the coiled DNA and the recoiled DNA. So kind of if they showed you the window inside, so here's how it uncoils, recoils, and then here's how the mRNA breaks off. And you see new freely floating RNA nucleotides coming in and lining up with complementary base pairs. So eventually it's going to reach a termination sequence in the DNA and stop making that mRNA. And then that mRNA is going to be processed. Let's see if I can find, it's the nucleosomes. Okay, see if I can find it. It doesn't look like it's on there. Okay, so we'll talk about these later. But so that's going to be for your transcription, and then translation is going to take place out in the nucleus after the mRNA is processed. So after you add the protective caps, the 5' G cap, the poly A tail, and then also after you splice out the introns and splice the exons together, it goes out into the cytoplasm and meets up with a ribosome at the start codon in the P site. The tRNAs are going to land in the A site, bringing the appropriate amino acids. And as that ribosome shifts down, reading one codon at a time, those that polypeptide chain of those amino acids is going to grow. And that is transcription and translation, otherwise known as the central dogma. So remember, as you go from genotype to phenotype, because proteins are how you phenotypically express the genes that are located in your nucleus and in your DNA. Whew, that's it. All right. Thanks, guys. And if you have any questions on any of it, definitely let me know at office hours or email me if it's after office hours or if it doesn't like match with your schedule. If you have something going on, email me your questions and let me know.